nice little group of people. I'm sure some other people will start trickling in, but we'll probably go ahead and get started. I just want to say hi to everyone and welcome to our live session today. I'm going to share a quick PowerPoint on who we are, and then kind of what we do as a program, and see what you guys are interested in if you want to join our sessions or if you want to join and help out with pre-health shadowing, and then we'll pass it along and kind of see what our doctor has for us. So we are a international student-led and women-led minority group. We're nonprofit and we're kind of here to help out pre-health students and kind of see how your journey is and how everything clicks together for you. And it's a fun journey, definitely. Just so that everyone knows, I will be going over this quick startup presentation and kind of introduce you to everything. We do have closed captioning, just in case someone needs anything. This is to accommodate all abilities and needs. And if you have any more ideas about how we could be a little better at being more accessible, you can email us at info@prehealthshadowing.com, or you can even just send a private message to me or Zywin, and you can contact us and see if you have any ideas. We'd love to hear them. So we'll continue. By the way, my name is Kira, and I am a volunteer and host at Pre-Health Shadowing, and I'd like to thank you all for coming. If you guys also want to stay in our loop and see kind of how our world works, we do have multiple social medias. We also have an email list. If you email info@prehealthshadowing.com, you can get on that email list, and then you can never miss a session. It also has information about upcoming doctors, but it also has kind of information on being a pre-health student. You can also follow us on social media. We have Instagram, TikTok, and X, which was Twitter. Apparently, it's been shifting around a lot. We also have YouTube, and then we have our website, but you can also find information on. So one of the fun things that we like to ask is, where is everyone coming from? I'm in California, Los Angeles to be specific, because there's often a lot of people from California. Let me see the chat. Oh, thank you for posting. Ooh, Kentucky. You know what's weird? I have not seen a lot of people from Kentucky. So they're one of my first little oddballs out there. So hopefully you're having fun. I guess a lot of people. Oh, New Jersey, Maryland. As I say, a lot of people don't want to say. You're all hiding. Michigan. Oh, 2 Michigans. Huh. A lot of East Coasters. New York. You get a lot of people from New York. There's a lot of pre-meds over there. So keep going. So if you're interested, I mentioned it a little earlier, that you can be a part of pre-health shadowing. You can also register to volunteer. It's completely remote, and it's also very flexible with your schedule. So if you did want to volunteer, you're welcome to come and see how your skills can kind of be a benefit for us, or also just kind of be a part of a community that uplifts each other and kind of helps you through the journey, because it's definitely a very hard path and a very long journey to get into med schools or to even become a PhD. That is a lot of work and a lot of effort, as I'm sure the doctor will tell you. I'll keep going. We also have something called a leadership team, and that's where it's 100% remote. It's a few more hours, so if you are looking to do that, it is a little bit more commitment than just joining as a volunteer. And we have a bunch of different roles you can apply today on our website at prehealthshadowing.com /contact/our team. Join our team, actually. And there's a bunch of different roles. I mean, if you're interested in grant writing or fundraising or video productions, we also have social media. You can work on kind of posting the videos or any of the ad copy that you saw on the website. We have a lot of people that kind of write all of the descriptions for our doctors. You can also head the volunteer department, or you can make up your own little position. We're always open to any ideas that people have, like if you want to do more website design type stuff, or if you want to look into more how to start more clubs at universities. That's also something that you can do. So we have a lot of wonderful opportunities for that. And if you want to play a bigger part in supporting it, I definitely recommend leadership roles with pre-health shadowing. We also have, if you're a high school student, I don't know if anyone is in high school, but if you are, we do have people in high school here pretty frequently. We do have a high school training program, HTP, and it's leadership and healthcare education. They actually help you in establishing your club on campus, but also give you materials and resources so that you can understand our mission and our goals and kind of how it would work with a high school program. So you're definitely not on your own with that. We do have a lot of people who are very happy to help you and kind of guide you on creating a club there. So if that's something that's interesting to you, you can always email us or go on our website. We do have an HTP tab on the website that will tell you a little more details on what the program is about and how you can kind of get started with doing that. So we will also promote diversity. We think that that's a key and it's part of our mission statement. And so to celebrate, we do host virtual discussions with groups and professionals to highlight important topics such as racial disparities, health, sexism, and education. We have a lot of people that tend to be more interested in hearing the differences and the kind of backgrounds to people in all kind of realms of pre-health. So if you do know someone and you want to nominate them, you can email us. We're very happy to kind of get those panels going. We haven't had one for a little while, but we are looking to start doing that a little bit more, just because we have a lot of people who enjoy learning about all the different ways of health and individualism. So if you do know someone and you want to recommend them, we'd love to hear from them and set something up. That would be amazing. So, as you know, we are free and completely open group. We do not take money or charge anything for these sessions. So we do ask that if you can, you humbly donate to our program because we are student run and we work around the clock so that we can make these sessions free, but also so that we can keep our Zoom up and running, our website up and running, all our resources open to anyone who can't necessarily afford to have all of these resources. So if you can, it would be amazing if you can just scan the QR code or go to our website, and even a few dollars helps. But if you can't and you know someone who can, we also humbly ask that you kind of message them and let them know about our program and our nonprofit. And that'd be greatly appreciated by all of us. So thank you so much for thinking about that. So throughout the session today, I would just drop all your questions in the chat. We will have co-hosts that are recording those questions, so don't feel as though you need to kind of write them down and hold on to them. We will have someone who's going to be making sure that they jot those down so that they get asked at the end. If you want to raise your hand at the end during the Q&A, you can unmute yourself when called on, and you can talk directly with the doctor. If you do not want to do that, feel free to message them and I can ask as well. So that'll be at the Q&A, the later half of the session. So take good notes. We do have a post-shadowing assessment at the end of this, and you will get a digital certificate verifying your hours on our course page. You just have to go and find the page that is responsive to this session. I'm pretty sure all of you have it because you joined the Zoom. So that should be pretty easy. We will be going over how to take the post assessment at the end of this session, so you can verify your hours. And lastly, we do ask that you turn on your cameras if you're able to. Always love seeing smiling faces, and it definitely makes us feel a little bit more connected versus staring at a blank screen. So we are respectful for any kind of circumstances. So if you do not want to put your camera on, you do not have to. So now I'd like to introduce Dr. Dhasarathy and say that she's an amazing environmental scientist and a geneticist, and she's going to go over her background information, her education, and then she even said maybe she'd introduce us to her lab, which is going to be exciting. So thank you so much for your time, and I would love to see your presentation.
Thank you so much for that kind introduction and for inviting me today. So let me see if I can first share my screen. And I'm hoping all of you can see that. Great. So this is my lab, and disclaimer, I'm not an MD, I'm a PhD. So what I do is more research. I also do teaching. So I'm at the University of North Dakota, and I'm at the North Dakota Medical School though. So I do teach medical students. I teach undergrads in the lab, and I teach graduate students. UND is, I think, one of the reasons it's most famous for the medical school is that we graduate the highest number of Native American and Alaskan Native MDs and physicians. So there's this in-med program called Indians into Medicine. And so a lot of the graduates are from the local tribal regions here. Okay, so a little bit about my background. Why am I here? So I was actually a zoology major, had a chemistry minor, and I had a biomedical genetics master's degree, both of which are from India. So a little bit different than the US. So I do want to introduce my students once I'm done, and they can tell you their career paths here, which would be more similar to what you guys are probably going to look for. So I came to the States in 1998, I think, yeah, um, with two suitcases and about $500 in my pocket. So I went into this PhD program at in biochemistry and biophysics at Texas A&M University. So yeah, I'm an Aggie, and I worked with Dr. Mike Kladde. He moved since I graduated to the University of Florida. So my degree was understanding, fundamental molecular biology and processes. After that, I moved to beautiful North Carolina. This is the National Institute of Environmental Health Sciences, and it's right across the street from, or across the lake. I don't know if you can see this lake here, a little lake. On the other side of the lake was the EPA building. So we interacted a lot with environmental scientists. But what we were fundamentally researching was, again, molecular biology of how genes turn on and off. And this was my postdoc mentor, Paul Wade. Following that, so big move from Texas to North Carolina, and now I live in North Dakota. We're about an hour south of the Canadian border. So you can imagine, so this little town called Grand Forks is where we have the one and only medical school in the entire state of North Dakota. And you can imagine that it looks really, really cold, and at times it does feel like we are living on another planet. But we get some other, this is a picture from campus. But in the spring, we do have all four seasons. So we get beautiful spring and summer and fall. So it's not always looking like another planet. What do we do in the lab? So you can see here a picture of a family, a really nice mom and dad and their kids. And the one thing that you may notice right away is that they kind of look like they are part of the same family, right? They look like their parents. And so this is the whole idea that children resemble their parents, or genetics. Everybody knows about this, and we know that each parent contributes one of each of 23 pairs of chromosomes, right? And what we are looking at is understanding how genes on those chromosomes are actually expressed. So if you think about a gene, it's like a recipe for pizza. Everybody likes pizza, I think. So this is a gene. If you think of a chromosome, it's like a cookbook containing several recipes. So each gene is a recipe. A collection of those recipes is a cookbook. So that's your chromosome. And then if you have a set of cookbooks, that is your entire genome, your 23 pairs of chromosomes, right? And so what we try to understand is the central dogma of the DNA in the gene is made into something called RNA, ribose nucleic acid. So this process is called transcription. And then the RNA encodes amino acids, which then end up becoming a protein. So this whole process is what we study in the lab. And that gives rise to a number of individuals. If you think about it, how many of the bases of DNA make up our genome? If you know the answer, it's about 3 billion. Okay, so if you write it out, this is what the number would look like. But how many of that do we share with every other person on this Zoom? We actually share 99.9% of our genetic material. So the differences that we see in our hair color, eye color, skin color, et cetera, height, weight, all of that is dictated by only 0.1% of our genome, which is still a lot, right? It's still about 3 million or so bases. But that's genetics. However, what we study is something called epigenetics, which means above the gene. And this has been a lot in the news lately about what this means to gene expression. And one way to think about it is these are changes that tell your genes which genes to be turned on and which to be turned off. For example, if you think about your entire body, right? You've got millions of cells. Every cell in your body has the same genome, the same 23 chromosomes. However, you still end up getting cells that are in your eyes, cells that make up your hair, cells that are in your liver or your pancreas, right? They're the exact same genome. There's the exact same genome in all of these cell types, and yet those cell types look very different. It's like if you take this famous Laker, and let's say he's 6'9", but what if you epigenetically change his genome? You would get several different kinds of heights for these people. So this is epigenetics. So they all have the same genetics, but because of epigenetics, how those genes interact with the environment, you can end up with a range of heights. Okay. So it's like taking this gene sequence, multiple copies, but then epigenetically modified them such that this would be expressed or made into that protein, DNA, RNA protein, whereas this one might only be expressed in neurons, but nowhere else, whereas this other gene was completely silent. Okay. And so it's like you can think about it as adding exclamation points, punctuation marks to a sentence. So instead of let's eat, grandma, you have that comma there. You might end up with let's eat, grandpa, or let's eat, grandma or grandpa. So just by changing the punctuation, not changing the actual words in those sentences, you can change how that whole sentence, what that sentence actually means, right? So to summarize, genetics refers to mutations. So you're physically changing the sequence that makes up that genetic word or phrase. Whereas epigenetics simply means you're changing the punctuation marks. So the gene is the same, the DNA is not changed, but how that gene is interpreted in that cell, by that cell, to produce RNA and protein or not to produce RNA and protein, that's what's different. So this is how we get from the same stem cells, you get all these different types of cells. So cell state A, we have about 23,000 genes in each cell. Cell state A, you have certain genes that are on and the rest are off, and you might get blood cells. Cell state B, you have a completely different set of switches that are on or genes that are on and off, and now you produce a neuron. So this is basically what we are studying. But how? How does this work? And so the answer to that lies in the nucleus of a human cell. So this is a picture of some cells, and the nucleus are these little blue circles that you see there. And that's about a 6 micron or 6 micrometer sized nucleus. Okay, that's about, if you think about it in terms of you pluck a hair from your head, 1/10 of that width. Now, if you zoom into that nucleus and you ask, what is the length of DNA within this nucleus? How much DNA is packaged into this, 6 micron size nucleus? It's 1.8 meters. That's long, right? It's almost 2 meters if you stretch it out. That's a lot of DNA. So how do you package that into that tiny little nucleus? So if any of you knit, you might know the answer to this. Or you know someone who knits. If you take a ball of wool and you stretch it out end to end, it's going to be very, very long, right? But then it's rolled up and rolled up and rolled up. Then you can stuff it into your backpack or your handbags. Same thing happens here. You're taking 10,000 miles of spaghetti and you're shoving that into a basketball. That's the analogy I can think of. So the cells solve this problem by just taking that DNA and wrapping it around proteins called histones, right? And that's great for packaging it. But if you want to turn certain genes on, you have to loosen that up to allow other proteins in the cell to get into the DNA, unravel it because it's double-stranded, and then make RNA, and then RNA ends up getting made into protein. So there are a number of really interesting proteins that we studied that are functional, and there's different types of proteins in each cell type that decide, okay, these genes need to be turned on, which means we need to unravel them, allow them to be made into RNAs. Whereas these other genes, we need to package them really tightly so they are not opened. And so you're not expressing a protein that should be in the pancreas, in the blood cell, or you don't express hemoglobin in your neurons by mistake, right? So this is a very, very tightly controlled and regulated process. And if any of these processes do not go as planned, then you end up with disorders affecting a number of processes. So that's how you get cancer. Development doesn't proceed as normal. So you end up with a lot of deformities in children. You can get obesity or memory issues or Alzheimer's and Parkinson's, neurons. All of these aging obviously happens to deregulate some of these processes. So this is sort of what we study. One example is if anyone has a calico, the reason why there's this patches of black and orange fur is because there are two X chromosomes. These are almost always females. One of the X chromosomes gets silenced by something called a non-coding RNA that coats the entire X chromosome and silences it. Randomly, sometimes the gene for the orange fur, that X chromosome gets silenced, and sometimes the black fur X chromosome gets silenced. So then you end up with patches where here the black gene coding X chromosome is silenced, and here the orange is silenced. So that's why you end up with those patches. Another example would be twins. They have the exact same genome, right? But one twin might develop asthma or cancer later in life. The other twin's perfectly fine. And then the classic example of epigenetics is this experiment done at Duke University, where these mice, they're called agouti mice, they were split into two groups. One was given a diet supplemented with additional methyl groups, and this one was not given any dietary supplementation. To their surprise, they found that this group that had like a lot of this nutritional folate groups had offspring that were mostly brown and healthy, whereas the normal no dietary supplementation mice group were just like the parent, yellow and unhealthy. What happened was epigenetic. Same exact DNA, same genes, but this group of mice had, because of the folate group, they were able to methylate their DNA, meaning they added the small chemical tag called the methyl group onto certain bases on the DNA, which silenced the genes that coded for this yellow coat color. But this called this agouti gene. But that gene was right next door to other genes that made this mouse susceptible to being obese and prone to cancer and diabetes. Whereas here, all those genes were silenced. So this is a classic example of how what you eat can affect or influence your genes, how they're expressed. Okay. Um, so the good news for all this, the bad news, of course, is if you have a bad lifestyle choice, don't do drugs, you can predispose your children, your grandchildren to acquire some of these diseases. But the good news is we can manipulate these marks because they are reversible and they don't affect the DNA sequence directly. Those can be changed in the lab. And in fact, there are plenty of these drugs already on the market. So what do we do in the lab? We have two sort of broad-ranging projects. One is on cancer. So we are understanding this process of how epigenetics drives or is the mechanism that drives cells to detach from that primary tumor. These cells need to change their gene expression states. They need to turn off genes that allow them to stick together, and they need to turn on genes that allow them to migrate and move. So that they can end up in other parts of the body, sit down, and then regrow. So this is the process that we study. That's one of our projects. The second project we have is understanding how epigenetics applies to Lyme disease. So this has been spreading a lot lately because of the warmer climates. And we never had ticks in North Dakota till a few years ago. They started appearing here. And Lyme disease often will result in arthritis, carditis, or affecting the nervous system. And this number is increasing every year. So we are trying to understand how this epigenetic changes leads to this Lyme arthritis. So I wanted to introduce you to my lab. So this is Jenna. She is actually from California. Caitlin's a member also of the graduate school. So both Jenna and Caitlin are in different years in graduate school. And Molly here is a lab technician, so she helps a lot with the day-to-day business of the lab. So I wanted to take you guys over to our lab in a little bit, and you can ask them all kinds of questions. But before I go further, are there any questions from you guys that you want to stop and ask before I move on? No. Okay, I'll keep going then. Okay, so what can you do with a PhD? I know many of you probably are thinking of MD programs, but there are some programs that offer both an MD PhD. It's a little longer. But if you are interested in a PhD on its own, let's say you change your mind, you don't want to be an MD anymore, there are a number of jobs that are out there, and each of these R&D in companies, manufacturing, QC. Some of my friends are now patent lawyers. So getting a PhD, you can go into patent law. There are people that work in the government in regulatory affairs. So all of these, many types of science, you could do basic research like what I do. There's translational. So these are more translating basic science into a clinical outcome. And some people just do clinical trials in companies that are looking at drugs and how those would affect patients. So there's a number of jobs, and there are multiple sectors, academic, which is what I do, but there's people I know who are in industry, who work in government, there's nonprofits that they work in. There's a lot of literature out there, but these 3 books are pretty nice and they're probably updated by now, but there's plenty of information out there to learn about careers in science. All of these have their own pros and cons, right? So it ultimately depends on what you want to do. If you like bench work, but you don't like all the other stuff, then you probably want to look more into this our research and manufacturing type of jobs. If you like organizing things, maybe you want to do clinical trial management. If you like to write, there's a lot of writing jobs out there. So just depends on what you want to do. Like I could never do a patent law job. It would bore me to death. But that's why I like to do what I like to do is talk to students. I like to do research. I like to be creative and think about science. So that's why I'm doing what I'm doing. There are some options for you guys. There are lots of this. My IDP is really useful in trying to understand yourself, your skills, your interests, and then help you plan for your career. The Office of Intramural Training at NIH has a ton of information. They have videos, careers, a lot of posts. So talk to people. That's the best thing I can think of. Yeah, so I'm gonna stop there and then see if I can take you guys on a little tour of our lab, if that works. And feel free to jump in with questions. So let's see. We are actually in the process of moving. So a typical research building, you'll see lots of corridors like this, posters on the walls with people, students usually present these kind of posters at conferences and seminars. So you'll see a lot of those on the walls. And this is my lab right here. See who's in. Okay. Hello.
Hi.
So Jenna is right here. So do you have, I think Jenna is going to talk to you for a couple of minutes about what she does. They also want to know career paths. So if you have how you got here.
Okay.
That would help too.
Sure. So yeah, my name's Jenna. I'm in my second year of graduate school. And my main research project is going to be studying a receptor that's implicated in breast and ovarian cancer. So we're looking into how that receptor functions and where it localizes to on the genome, specifically in ovarian cancer cells. And a little bit about how I got to UND. In my undergraduate, I was fortunate enough to get placed in a lab and get some research experience. So as an undergraduate, I did bench work for about 2 years, where I was able to get some basic lab skills. And I was also fortunate enough to be in a program called the URISE program, which is through the NIH. And that's specifically for students who have the goal of pursuing their PhD eventually. So if you already know that that's your end goal, I would highly recommend looking into what's required for that program. It's a great program. And then, I applied to UND. I actually spent a year as a lab technician before getting into graduate school. So that allowed me to further bolster my CV and the lab skills that I learned in that time. So I guess the biggest priority, if you know that you're interested in, well, I guess you guys are pre-med, but still you need to get your research experience. So just kind of prioritizing that, not being afraid to reach out to people and professors. There's plenty of opportunities if you just seek them out. So, yeah.
I have a quick question. What are you using to study the receptors or look into that? Like what kind of technology or machinery?
Yeah, so I guess studying this specific receptor, it's called estrogen receptor alpha. So there are specific antibodies that have been generated that specifically recognize this receptor. And so you can use those antibodies in various assays. So like some of the assays I'm doing look at estrogen receptor alpha binding to chromatin. So like within our genome. So we'll basically isolate DNA out of these ovarian cancer cells, and then we'll use that antibody that's specific to this receptor and see where on the genome this receptor is specifically binding to and acting upon. So that's kind of where the specificity comes from. And there are antibodies generated for all sorts of proteins that are studied. So.
Oh, and what would your end goal be? Is this more treatment or diagnosis?
So, yeah, I mean, the end goal is mainly to find out more information. It's not, so sort of the background for this project is that in breast cancer, estrogen receptor alpha is very successfully targeted with chemotherapy. and patients do see a reduction in their tumor size and can even go into remission. There's a chemotherapy called tamoxifen that is extremely successful. It targets estrogen receptor alpha. However, in ovarian cancer, this same drug is not as effective, and it's unknown why. So even though estrogen receptor alpha is expressed in ovarian cancer, it doesn't behave in the same way. It doesn't respond in the same way to these chemotherapies. And so that's sort of the basis. We're really just looking for information. A lot is unknown actually about this receptor in the niche of ovarian cancer specifically. So really it's just gathering information, what's different about it in ovarian cancer? Where is it binding? What are its binding partners? Just sort of gathering that information. And then we can go in a more targeted direction based on what we find.
Wow, that's amazing.
You got a question in the chat, I
Oh.
think.
Yeah, you did, but
What's been more surprising or encouraging about cancer and Lyme disease research? Well, the cancer research nowadays, I think one of the biggest leaps forward has been the huge amount of improvements in genomics research. So we do a lot of, instead of looking at one gene at a time, we can look at multiple at the same time. And those technologies have gotten cheaper and cheaper. So now we can really query genome-wide very quickly. Also, I think some of these genomics tests are cheap enough where some clinical applications also started. So like as a patient in the next few years, I think it wouldn't be very surprising to see some genomics assays used for testing. So I think that's been one of the most encouraging aspects about genomics research. With our Lyme disease research, before we started looking into this, no one knew anything about epigenetics being linked to an infectious disease like Lyme disease. So I think I was kind of surprised that no one had looked. And so actually, this is a perfect time for me to bring you over to Caitlin, and if she can spare a second.
Yeah, absolutely. I didn't
Sorry.
there.
Then she can tell you about her Lyme disease work here.
Hello. So I'm Caitlin, as I was already introduced, and she probably told you a little bit about the Epigenetics of Lyme disease project, but another cool project that I have that's kind of fun to talk about is we're creating an organoid of Lyme arthritis. So what an organoid is it's a cell culture that mimics what you would see in a human being. So if you think about Lyme arthritis, that's arthritis in the joint caused by the Lyme disease bacteria. So for an organoid for our Lyme arthritis, you'd want to have all of the same cell types that you would have in a joint. So like in a joint, you have the cells that make the synovium, the synoviocytes, you have bone cells, chondrocytes. There are immune cells in the joint as well. You might not have known that. So another cool project that I get to work on is I'm attempting to create this organ in a culture dish so that we don't have to rely on mice and human beings to do research. And then, of course, like Archana already mentioned, there's also the Epigenetics of Lyme arthritis project. Yeah. Do you have any questions for me?
I have a question. Have you ever worked with like 3D printing tissues, or how are you creating?
Yeah, so we actually have a partnership with the Biomedical Science Department, and they print the bone scaffolds for the organoids. So I actually have one of them here, and I can try to show you. So if you look at it, it kind of looks like a Chex Mix, but that's the bone scaffold that the cells grow on. So yeah, we're trying to mimic all of these structures you'd find in the body in a culture dish to help our experiments have more accuracy.
Awesome. How are you determining like progress in that? Like, do you have some kind of like UV or X-ray or
It's actually pretty simple.
microscope?
So the first step, right? You just want to make sure your cells are living. So like the first thing we do is we quantify like live and dead cells. Do I add them to the culture dish? Do they die? That's bad. Like need to reassess. And then kind of after that, what we have is data from mice. So I know that in mice, she talked to you about the central dogma. In mice, certain genes are expressed. I can look at the gene expression of the cells in the organoid, and then I can match it to the mice. And then if it's done correctly, I would assume that I would see the same things I saw in mice as I saw in the organoid. So yeah.
Is the bacteria for Lyme arthritis kind of similar to other behaving bacterias, or is it different in any way?
It is pretty different. So it's a spirochetal bacteria, and the interesting thing about it is it is not adapted to survive in people. It's not meant to be an infectious bacteria for human beings. It's meant for ticks specifically. So it's adapted to survive in the tick midgut. So kind of like the interesting thing about it is it doesn't have a lot of the same virulence factors that other bacteria would have because it's adapted for ticks, not people. So when it gets into people, it's kind of confused and goes to these few specific regions. But yeah.
Interesting. That's really cool.
Yeah.
Well, thank you for that.
What are some of the other regions that it affects? You mentioned arthritis in the joints, but are there other areas of the body that
So I specifically study Lyme arthritis, but there's also Lyme neuroborreliosis. So that is the bacteria get into the meninges in the brain. There's a coating around the brain that the bacteria have been known to get into. There's also Lyme carditis. So they interfere with the cardiovascular system and cause inflammation in the heart. So I don't know if you guys are familiar with long COVID, how they saw effects in all these different systems, but it's a little similar with Lyme disease, where these people that get Lyme disease have all of these systems infected by the bacteria. So it's not everybody that gets Lyme disease gets Lyme arthritis, Lyme carditis, not Lyme borreliosis, but some people do. So yeah.
Thank you.
Yeah.
Any other questions? Good. All right. So this is a typical lab setting. Some of it's messier than others, but that's okay, I guess. That means people are working, right? Here's Molly. So Molly's our lab tech. She's in charge of, she's actually planning to go to medical school, so she can probably talk to you a little bit about that.
Yeah, she said I'm the lab tech, so I do all the ordering. I kind of keep this place running a little bit. I keep stock of everything. I organize everything, and then my main project is on this protein called Spark expression in bladder cancer and its implication in the severity of bladder cancer, as well as how it is regulated. I think she mentioned methylation and transcription factors that we are looking into to see how this Spark is being regulated in bladder cancer. And then, yeah, I'm working towards applying to medical school.
If you went to medical school, are you going to still look into Spark, or are you going to shift into some kind of different research? Is there anything you kind of have your eye on?
I would, if I was continue research in medical school, I'd probably just stick with with this, and because we can only do so, like within one paper, we can only look into so many things, but I could expand upon it more if I had more time.
Are you planning on going to oncology then?
Yeah, I was thinking oncology as an option for that. Possibly Ph.D., M.D., so I could do research and practice medicine, but yeah, we'll see.
Well, that sounds awesome. Definitely working in a lab helps you out with your MD, PhD route. I've heard that is a brutal route to go down.
Yeah, but research experience, that's like why I was recommended by another professor to work in here for research experience, and that's how I ended up here.
Would you say that I know you're not in it yet, but MD, PhD route is centered on more PhD route stuff or more MD stuff, or is it a 50-50? Have you looked into that at all?
It depends on where you go, and because some schools will do it differently, because I know at least at UND, they start you off mostly MD, and then you eventually shift to more the PhD, your more research aspect, but it depends on the school.
Sounds good. Do you have any schools in mind?
UND for sure. I was also thinking University of Minnesota. No warm place. Yeah, no, I can't go anywhere too warm. raised in North Dakota, so I can't, yeah, somewhere up north.
Sounds like a nice plan. Does anyone else have any questions? I think we're good.
I don't think so.
No.
Nope. Okay, well, thank you, Molly.
Thank you.
Yep. Thank you, Caitlin and Jenna. Okay, so I can head back to my office. Any questions I can answer in the meantime?
I had a question. What does your kind of typical week look like as a PhD?
For a PhD student or me specifically?
You specifically.
Me?
Yeah.
It's very different. Not bothering someone else. Every week is different. And I think even every day seems to be different. So my time is a mix of research. So I actually still do some bench work. A lot of faculty don't anymore, but I feel like if I don't do some bench work and have fun on the bench, I get really grouchy and nobody likes that. So I still try to keep doing some bench work. Another thing we have to do is write grants. So there's a lot of writing involved, which I did not sign up for initially, but now I've gotten used to it. That's how we get funding to run the lab. That's how we get money. There are meetings. So I'm in charge of a couple of different groups on campus. So that means I have to coordinate meetings and talk about progress on whatever project those are. So there are some of those meetings every other week or so. I teach, so I work with undergrads at the bench. I have medical student teaching, so I do cases with them. So they're given a case report and they have to sort of understand all of the symptoms the patient has, what type of test they would order, et cetera, and I sort of help guide them through that. And then let's see, grad student, I do some teaching for graduate students, molecular biology. These happen at different times of the year, so I'm not overburdened all at the same time. So for example, in the spring, I do the medical student teaching. In the fall, I do the graduate student teaching. So that's my typical week, I guess. It's a mix of teaching, meetings, bench work, grant writing, paper. I write and publish papers. So there's a mix of everything. So you have to be a little bit you should be able to juggle a number of different things because your writing brain is different than your bench work brain and your
Definitely.
teaching brain.
I know you said that some people aren't doing bench work. Are you kind of flexible in not doing bench work, or is that something that at a certain point you have too much kind of
Yeah, you have
administrative
too much
roles?
going on and
Yeah.
you're like, I don't do that anymore. Here, you do it. So yeah.
How is it with finding people in your lab? I know that some people in here that might want to be looking for a lab. What do you think that kind of makes them stand out as someone who potentially could be going into a lab?
So our graduate program does it. I mean, every school has its own unique graduate program, right? So some schools they want you to contact individual professors, talk to them, and then make sure that they have space, and then you apply and you say, I've already talked to Dr. So and so to join their lab. Our school's program, at least in the medical schools, biomedical sciences program, they do it a little bit differently. So what they do is you apply to the program and you want to say that, you looked at do your research, look at the web page and say, I've looked at these labs and these labs are what I'm really interested in. We have a very strong neuroscience group here. For example, we have a very strong epigenetics group. So you may want to look into that and say, I want to look at Dr. A, B, and C's work sounds the most interesting to me. Once you get into the program, they do what are called rotations. So they spend 6 to 8 weeks in 3 different labs. So they do 2 labs in the fall and 1 lab in the spring semester, maybe slight overlap. And at the end of those 3 rotations, they select the lab that they're going into. So this gives you guys, if you are interested, that gives you a little bit more flexibility. You may really want to go into one person's lab, and then you get there and you find, oh, I hate this work. I don't want to do it anymore. So whereas you might find another lab that you never considered, you might find their lab very interesting. And that happened to me. So I can tell you that happens. So this really helps. But what would make a student stand out is, number one, do your homework. Look into what that lab does, read up about it. You need to clearly state why you're interested in what's the problem that you're interested in solving, because PhD is about solving a problem. Read up a little. You don't have to know every single detail, but you should at least be aware of what the lab research is about. And then contacting people, go and talk to them. Even if you don't want to get to that university, you can always email the professor and say, I'm really interested in applying, and I'd like to know more about your work. Would you spare 10 minutes to talk with me on Zoom or on the phone? And people would be usually willing to do that. Do not just apply to them and say, I want to join your lab. I get like, I don't know, 50 emails every year saying that they absolutely have to join my lab. Sometimes they send me CVs that have nothing to do with what my lab does, and that's okay, provided they can explain why they're interested in my lab. You'll be surprised at how many times that does not happen. They start talking about, some people start talking about their expertise, and then they say, I'm really interested in your lab, and can I join your lab? And I'm like, why are you interested in my lab? So you really need to tailor your emails or your resumes or your CVs or your letter of intent, whatever it is that you're submitting, to that specific university or to that specific professor. I've gotten emails from people saying they want to join my lab and they have the wrong name. So instead of me, there'll be somebody else in the department they were emailing, but it came to me. So I'm like, I don't work on that. I don't work on this at all. Why did you send it to me? So please be really careful when you apply to positions like that. So that's my
It's funny that you mentioned that, because I know that a lot of people go into med school and they have this clear defined concentration they want to go into, and then they end up in it and then they go, you know what, I don't really like this.
Yeah, exactly.
And they end up in a complete other concentration they didn't even consider.
So one way to avoid that is to get a hold of people that you know are in those fields, and you call them up and say, what do you like about your job? And specifically, what do you dislike about your job? And then that will give you an idea of whether you want to do that or not. They're called, by the way, there's a name for this. They're called informational interviews. And I know you had mentioned a number of, Twitter and all that, but I would highly recommend LinkedIn as well, because a lot of people who are professionals are on LinkedIn. That's more professional. So please don't post like pictures of your cat and dog or something, but make a very professional resume. And if you connect to other people that like your professors, for example, they might have contacts, maybe people that they went to school with who are now doing exactly what you want to do, who have your dream job. And LinkedIn is a really nice way to make those connections. And then you can directly contact them through that app and say, hey, I'm so and so student. I saw that you work in, I don't know, this industry. I would really love to learn more about your job. Do you have 15 minutes to talk to me for an informational interview? Most people are okay with that because you're not asking them for money or a job. You just want 10 minutes or 15 minutes of their time, and then ask them very pointed questions. Or simply email and say, I want to know these things. If you don't have time to talk, could you send me a short email back answering these questions? That might be another way to do it.
definitely some great advice. I've definitely gotten more and more into LinkedIn. There's a large presence on there. And
Yeah.
pre-health shadowing is on there. We
Oh,
post things
good.
all the time,
Yeah.
which we
Good.
love to do.
Yep.
With your heavy workload, do you do anything fun outside of that?
Oh yeah, I try to anyway. It gets harder and harder to do. But I have both a cat and a dog, so I spend a lot of time taking my dog for walks. There's a lot of like trails around here, which is one of the nice things about being in a smaller town than I'm used to. Um, so yeah, I do a lot of that. I love to cook, so I always try to find some fun new recipes to try out. Um, I played the guitar. Don't do a lot of that anymore, but I try to every now and then.
And instruments are always hard to keep up with.
Yeah.
What do you think when you're going into this
Mhm.
was your kind of biggest pitfall? Like, what did you not expect going into your PhD?
Yeah. So this is something that I never thought of. So PhD, when you're an undergrad, you're like, oh, cool, research. This is so fun. I get to find out new things, right? Um, your PhD, you start to realize that not everybody knows the answers, right? If they knew it, then we wouldn't really need to do any research. So there are questions that you get to ask that nobody knows the answer to, which is exciting. But there's two things about that. One, you have to struggle quite a bit to get to the point where you're like, okay, now I have somewhat of an answer to my question. Your PhD mentor doesn't know all the answers. It's up to you to sort of work your way to that. So that was something that I didn't expect, right? Um, Naively so. The other thing is all through my PhD, all through my postdoc, which is sort of like you spend a few years after your PhD sort of working on different projects to get more experience and also to define a new question that you want to answer as an independent scientist. Both those times you're heavily trained as a bench scientist. As a faculty member, you get to do very little bench science. After the first 2 or 3 years, your job is to train others, and then they kind of go off and they're focusing on the bench work. Your job is to write grants to get money to do the work. So that's the part that I was not, I mean, I kind of expected, but I didn't realize how much more work you have to deal with. Running your own lab is like running an independent business, which means you need to work on the budget, you need to write the grants to get the money, you need to get the work done so that you get more grants and more money. You need to be able to write. Writing publications is very different than writing a grant. And now with all the craziness happening, I don't want to get political, but with all the craziness happening with NIH funding, that's very, very stressful right now. So these are things that, are beyond our control, but things that you don't expect to happen.
Yeah, I can definitely see that. Um, I had a question, and now my question has kind of flown away. Oh, I was going to ask you. You seem like you're doing a lot, like tons. How do you kind of navigate burnout? Either not getting burnout, or if you're burnt out, how are you kind of navigating getting
It's
out
a
of that?
it's a huge problem. And I think the biggest burnout times are, I'd say, your first few years as a new faculty member, right? Because you are trying to get tenure, which means that they can't fire you unless you do something really crazy. So that tenure process is extremely, extremely high pressure. So it's difficult not to get burned out. So after I got tenure, I kind of took it down a notch because I'm like, I really need to recharge and recover before I start. And then we got COVID. So um, it's really about knowing your boundaries, learning how to say no. That was a big thing for me, and I know it is a problem for a lot of female faculty, is learning how to say no because they try to dump a lot of things on you that somebody else should really be doing. Um, you get better at that. Other than learning how to say no, I've made certain rules for myself. So, like Thursdays are my writing day, and I will not take any meetings. So I get uninterrupted writing time, which helps my productivity, and that way I can turn my computer off at 6:37, go home, and I don't have to think about, um, work for a little bit. So take my dog for a walk. So it's just learning that the realization that you cannot do everything 100%. So learning how to sort of say, okay, that one I've done 80%, that's good enough. Let's send it off to the next person. So just learning how to balance your time, and it's still hard, it's still a challenge, and I don't have all the answers, but I just found some things that worked well for me.
I like that answer. I've definitely been on and off of the burnout role. So
Yeah.
I always love strategies and kind of ideas on how to fix that or kind of solve that.
But
Is that
another thing I found is if someone tells me, can you do this one more thing for us? I would say, okay, I will do that if you tell me which item I can take off my to-do list. So then
okay.
like my chair might say, oh, you're doing that other thing also. Okay, don't do that. I'll get somebody else to do that for you, and you do this for me instead. So that has helped.
Okay, I like that, switching goals.
Yeah. Sorry.
Is there any kind of condition, or I know you're focused a lot, or your lab is around cancers. Is there any other kind of conditions that you look forward to researching?
Right now, we have our hands full with that. But, the nice thing about working in epigenetics is that it's the same mechanism, it just happens a little differently in each of these conditions, right? So, one of the things I'm really looking forward to working on with a collaborator is there's an Alzheimer's disease project that they really want our help with, a colleague of ours here. So that's something where we haven't done anything yet with them, but that's something we may get into the future. There's a group here who is looking to get into space biology. So understanding how epigenetics changes happen to astronauts go into space and back. So that's something I thought was really cool and wanted to look into.
When I was little, I used to love the idea that you could go into a black hole and you could come out like a foot taller.
Yeah.
Well, that probably isn't accurate. It was always a hilarious idea.
Yep.
Do you think there's any kind of future goals to mix like immunotherapy and epigenetics, or
I
kind
think,
of have that like mixtured
yeah. So there
research?
are a lot of what are called epi drugs on the market.
Mhm.
And one of the things they have found is that certain drugs, when given in combination with other drugs, make the cells more sensitive. So by targeting certain epigenetic proteins, so you give them the epigenetic drug first, um, that targets those epigenetic proteins, then you give them the other drug, which normally wouldn't work in a patient, say an immunotherapy drug. Now you make that cell more susceptible to being killed by the immunotherapy drug. So there are some studies out there where they're doing these sort of combination therapies. So yeah, there's definitely that option.
Yeah, because I've noticed that there's a lot more like mixed degrees now in
Mm,
MD routes where you
yeah.
can do like neuro and psych.
Oh,
Um,
yeah.
so I was kind of interested in seeing if there's a connection there because I know that there's certain kind of overlap between immunology and epigenetics a little
Oh,
bit.
yes, there
Um,
is. Oh, I see what you're saying. Yeah, no, there are plenty of people who study the epigenetics of immune system and the problems associated with that. So yeah, that's known for a while now.
that's very fun. What would you say is your favorite part of your job or your PhD route?
My PhD route or my current position? For PhD, I think I was just so excited about learning all these new things. And, I did my PhD during the genome revolution, so I could see hands-on. Once, as soon as I started my PhD, they sequenced the human genome, which I was like super excited about. So it was kind of fun to live through some of these breakthroughs. Um, and as currently the best part of my job is really hanging out with the students that you just saw or you just met, and they make my day. I come back from a meeting where somebody's being disagreeable, and I come in, and they're like, "Hey, you want to see my data? Look at this. See what I found." And that really makes my day. So that's, I think, the most fun part of me having this job.
That's awesome. I know that you listed a few books earlier on kind of different careers in science. Do you have any favorite books in general of whether it's research or life or kind of any of those?
So those three books are specifically, I think it's in the PowerPoint I sent you guys. Um, those are the three that I know are probably some of the best for looking at careers in science. Um,
I meant more in general, like,
in
do you have a
general,
favorite book about life or like a favorite book to study or like life goals or career goals? Those
oh,
kind of
I
books.
I don't actually. I haven't read a
I'm sure
whole
you're busy.
number of career-ish books. Other than those three, I just wanted to know what other careers in science there were. But I read so much scientific literature at work that I'm mostly reading like fun, reading for fun at home.
I know a lot of people ask questions about mentorship in previous sessions. And I noticed that you had mentors, and I'd say the top two questions are always like, how do you find your mentor? And what do you think like the top benefit of having a mentor is?
Yeah, so these are my mentors for PhD and postdoc. That's whose labs I worked in. Um, but I think I attended this career meeting recently where they said, in addition to a mentor, what you really need is a sponsor. And so mentors are different, right? So for me, my mentor was, I really liked so and so's research, so I contacted them and, joined their lab, and they were my scientific mentors, right? I also have other people that I go to for mentorship who are not necessarily scientific mentors, but advise me on, oh, I have this, hey, I got this offer to go to this other place. What do you think about it? What should I be asking? What are the questions I should be asking? Is this a good career move for me? That kind of, someone who gives me career mentorship. Um, a sponsor is someone who is more than just a mentor. And this is something I heard about recently, is it's more important, it's also important to have a sponsor. A mentor is just going to give you advice, right? A sponsor will actively uh, help talk about you to some other people and promote you. So, for example, um, what is a good example I can give? Um, Somebody who, let's say there's a singer who's not very well known, but they talk to, I don't know, Dolly Parton. And so Dolly Parton knows someone who's organizing a music festival, and she goes and says, hey, you should talk to this person. She's a very good singer, right? That's sponsorship. So in this situation, it would be like me talking to someone, say, hey, I have a student who's fantastic. She needs to talk to you guys about her research, right? So sponsorship, someone who sponsors you, I think would be it's a good attribute for a mentor to have.
Yeah, I've never heard of a sponsor. I'll have to look
Yeah,
that up.
it's a new term.
Did you ever have doubts going into your PhD?
All the time.
Oh.
That's also something they talked about. It's something called imposter syndrome, right?
Mhm.
I don't know if any of you have heard of that. So I was kind of shocked to hear at this meeting that the person who gave this seminar was the uh, dean of a medical school, and they still have imposter syndrome. They still doubt themselves. So, if any of you are undergrads or high school students, I'll tell you this right away, it never quite goes away. And this lady said the exact same thing. You will always have doubts, but the thing to remember is do it anyway. You may have doubts, oh my God, I'm never going to get a PhD. I'm never going to get into a PhD program. Do it anyway. Oh my goodness, I'm never going to get a postdoc. Do it anyway. I'm never going to get into an MD program. Do it anyway. You just have to try. Um, and the other piece of advice I heard from this person, which I thought was really funny, was she said that the voice in your head that tells you, "Oh my goodness, I don't like, I have all these doubts about this. I don't think I'll be a good, you know, MD student, whatever." She referred to that. She said, refer to that voice in your head that casts these doubts on your ability and you, as give it a name of someone that you don't like. And she used the example of, you guys remember Ursula from The Little Mermaid? Yeah, that's Ursula. Ursula, shut up. So you just have to keep telling Ursula to shut up and go away, and you go and do it anyway. So that was,
that.
I thought, a really good piece of advice I picked up.
That's definitely great. So we're kind of wrapping up. Do you have any other pieces of advice that you'd love to give people who are either going PhD or MD or maybe doing the combo? I wish you guys luck if you're doing the combo.
Try and find out from if you know people who have gone the same path that you have gone or you want to go on, try and talk to them. Um, and talk to people, try and figure out if they know anyone who is in the position that you want to be at one day, get their advice. But at the end of the day, think very hard about what you want to do. Both of these paths are very difficult to do. And I've known people who've gotten into an MD program, who got into a PhD program, and within a year or so realized, oh, this really is horrible and I don't want to do it anymore. So really try and understand what that involves, the amount of work it involves, the amount of commitment it involves before you go into it. That being said, after you start and you realize maybe a year or two in that you don't want to do it anymore, and you're 100% sure you do not want to do that anymore, that's okay. The sooner you realize that and you change, the better it is for everybody. And I've told this to PhD students. We had one who was like just sort of hanging on because her family really wanted her to do it and she did not want to do it. And we said, you know what? Don't do it. If you don't want to do it, you only have one life to live. Go do something else. Go do something that makes you happy and will give you a career that you want. You don't have to force yourself to do anything. Um, so that's my piece of advice.
I love that. I always used to say 2 cents, and apparently that kind of devalues your statement. So I love your $1 million. And I also definitely agree with find your passion because
Yeah.
I like that phrase of do something you love because you'll never work a day in your life.
Yes, exactly. Yeah.
So I will wrap up with a mini presentation, and I do want to just thank you so much for all of your information and letting us kind of go into your lab. I think that was very special. I have not been able to do that online, and so I was very excited to see it kind of. And you even introduced us to all your students, which was like
Yeah.
extra special.
Yeah.
I know that some people will just kind of show some pictures or maybe they'll show you a little video they took, but it's definitely a lot more special to be live and in person.
Yeah, I feel like you need to hear what they do from their own mouth because they're both really, really cool students and Molly's really fun too. The undergrads were not here today, otherwise I would introduce you to them, but they have classes and stuff, so they couldn't be here.
Busy lives as researchers. Well, I just want to say I appreciate it a lot, and I think we all do, and we all learned a ton. I know that Geraldine over here is thanking you and saying she loves her
Oh,
your "do it anyway" quote. She's probably gonna keep that memorized because I know I
thank you,
am.
Carolyn. And if
So
anyone wants to chat or get more information, please email me and I'm happy to answer any questions. I leave you with that.
Awesome.
awesome. I'm
Thank
sure
you so
some
much.
people will take you up on that.
Okay.
So I do want to just submit our little wrap up and kind of explain to you guys what to do post session. We do often recommend that you guys have a little reflection. You can write this on your own. You can write it on LinkedIn. You can tag us. Sometimes we'll repost if we love what you wrote. And you kind of took the time to have a thorough reflection, pros, cons, whatever. We do have the kind of three questions if you're stuck on what you're going to reflect on, and it's what brought you today, what are your three major takeaways, and what do you want to learn more about. I also like putting, like, if I was going to do my own interview, what questions would I ask? And then you can, if you ever decide to do an informational interview, like Dr. Dhasarathy was saying, was you could oh, you could just have your little list because you already kind of thought about them post session. So that's not required, but if you do want to share them, you can share it on LinkedIn and you can tag us. And we do oftentimes love kind of sharing all of the involvement in the tagging for our professional network. You can also just email it if you don't have a LinkedIn, and we can still tag like your name. And then if you ever get LinkedIn or you want to establish LinkedIn, your name is there, and you can kind of go find that and put your name into that. So that's always happy. I always love seeing everyone's reflections. If you're interested again in being part of Free Health Shadowing, we are 100% remote, so we're very easy. It is very flexible. You kind of pick your hours and you kind of pick what you want to do, which I absolutely love because I like the flexibility of I want to do this and that and this and that, and I don't have a fixed role or a set kind of job duty. It's kind of what best skills do you have and how do you fit in with the community of all these pre-med, which I love. We are mostly asynchronous. There are opportunities at times where you can meet with certain people if you're nearby. Those are pretty cool. I have not been able to do any of those, but a lot of those are on the East Coast. So if you're on the East Coast, you're benefited. So we are accepting applications. You just go to the website, and it's in the Get Involved section. I don't know if you can see my mouse, but there's a little tab that says Get Involved. And then that will tell you what you can do, and you can apply to the leadership team. Once again, we do ask that if you can donate, that you do donate. We are completely free, and so we run off of the time of our volunteers and donations. And so we are very excited when anyone donates even a few dollars. And if you can't, we always love word of mouth, sharing, not even just so that people can donate, but people can get more informed. And we always love our growing network and the amount of people that put their time and energy into this program so that everyone else can benefit from it. While we are everyone's competitive in the med school world, it's kind of nice seeing everyone come together and kind of share resources and topics and advice, especially if you've been in it or not. So now the easy part, I guess it's not too easy, but the easy part, are earning your digital degree for attending. It is timed. I don't know if that says on there, but it is timed. It's 30 minutes. You get 2 attempts to get a 70% or higher. The questions are not too tricky. If you sat in and you paid attention and you took notes, then you should be perfectly fine. I would not stress any of these quizzes. For if there's any kind of mess up, mistake, there's a typo or something happens, just feel free to message us, and we can always sort that out with you. That's not a big deal. And our email is info@prehealthshadowing.com. You just sign up for the course. You did that to get this Zoom information, so that should be an easy thing to find. Our quizzes are open indefinitely. You can actually go on our YouTube or on our website, and you can look at videos from years past, and you can still take the quiz and get the credit for those videos. It's always nicer to do the live sessions. So I do recommend you get on the emailing list, or you check in at the beginning of every week on the website and see who's hosting a session or who's going to be speaking. You can always find amazing topics and amazing people. I know that the doctor here was amazing. She was definitely one of my favorites. And then you can hit finish course. Some people mess this up. So when you finish your quiz, you actually have to go to the top and hit finish course. And then that will give you your certificate. Some people just finish the quiz and it gives you your score, and then they don't know what to do after that. So there's like a little finish course button at the top. And then you should be able to get your little certificate. I do say that our quiz will probably be up in 30-ish minutes. It's not usually up like right at the end, but we do a very well job of trying to get it up as soon as possible. But I would give it 30 minutes before you freak out and say that it's not working. And then our recording will also be added to our YouTube and our website. That's usually within the next 24 hours, just because it takes a while to download. So if you missed any part of this session and you want to go back to that, you can go to our website or you can go to our YouTube. The YouTube also has every single one of our sessions. So if you have a specific specialty that you want to get involved with, or if you want to watch a specific topic that kind of highlights what you want to go into, it's a great way of kind of seeing if you like that topic. And then you go to med school and you don't switch your topic. I know that's a frustration that people always have. So if you want to go watch any of those are all free, and our website, they also are free. So don't worry about trying to make some account or do any of that. It's hassle free. And we have sessions next week and every week. I know that our next session is on Wednesday, and that's with Dr. Barton Goldsmith, and he is a psychologist, so he should be fun as well. And I know that there's a few doctors coming up as well that are very fun and interesting, and I look forward to seeing them as well. So if you have any questions, feel free to stick around. Don't forget that we also are on social medias. We're on LinkedIn, X, which is Twitter. I'm never going to get used to that. Instagram, YouTube, and our website. Our website always has everything up to date. Our social medias are always up to date, but our website is always the best way to get kind of scheduled. You can also go, and for the next few weeks, you can go and you can just join every class that's coming up, and then you'll get notifications, and you can also add that to your calendar so you don't miss those. So I'd like to say thank you for joining today, and stick around if you have any questions. I'm happy to answer anything about a program or anything about pre-health stuff or pre-med stuff. I'm in that world now, so. And I also just want to say thank you again.
Thank
For all
you.
your time.
All right, I'm gonna log off unless someone has questions for me specifically.
Any last minute questions?
No. Okay.
No, I think we're good. Thank you so much again.
Thank you.
Have a good one.
Bye.
Hey, I just wanted to double check. Where do you reference the